All right. Thank you so much to Wendy and Orkin for giving me a chance to share a little bit of my work with you. I am local here, and I want to tell you a little bit about really cool soil bacteria, Myxobacteria. I'm coming from the field of biochemistry, so I'm a structural biochemist, but I ventured into Myxobacteria more recently because of the interesting biochemistry that they have, and I want to share that with you today. So, this is an outline of my talk since I don't have so much time. You may notice a common theme that light is very important in the work that I do. So, Myxobacteria are non-photosynthetic, but they sense light and they have molecular mechanism to do so. And they also have these special light-regulated enzymes, which is really fascinating to me as a biochemist to initiate chemical reactions by light, which is an abundant and free energy source that we have in our environment. So most recently, we wrote a review with my colleagues that was featured in Journal of Bacteriology. So if you're interested in Myxobacteria, and I hope you will be, they form these very unique multicellular structures known as fruiting bodies, which I also showed on my initial introductory slide. These fruiting bodies grow in the direction of light. They're multicellular structures, and they're unique to Myxobacteria. They were actually mistaken for slime mold when they were originally discovered. So these unique fruiting structures are assembled over course of time from vegetative cells, as you see here. So the vegetative cells come together, they form these mounds. The mounds can move actually along the surface of the agar, and ultimately they form these fruiting bodies. So this is a top to bottom view on JBacT's cover page that was featured. In addition, Myxobacteria form spores, and spores are extremely dormant, so they can survive for many, many years. And then when conditions are favorable again, the vegetative cells will form. So I come to you from the side of biochemistry. So I was very interested in light-regulated chemical reactions that are catalyzed by enzymes. And nature has evolved this beautiful system, and we were able to solve the cryo-EM structure of this wonderful enzyme, histidine kinase. So this is the enzymatic domain, and then this is a photosensory core module where the light is being sensed. And the reason I'm showing you the structure of this enzyme is because the light is sensed and travels over 120 angstrom along this long helical interface to trigger enzymatic domain. This photoreceptor senses red and far red light. And as you can see, we have a clear distinction in how the fruiting bodies form, whether the far red light or red light is being sensed. So we knew that we are onto something here with this fruiting body formation. This was the first view of light-regulated enzyme from Myxobacteria, specifically the enzyme that is triggered by light. And you can see the phenotype of the fruiting bodies on the side. Now, most people study Myxo for their natural product synthesis. So they have very complex metabolism. And when they're in soil, they're sort of a good citizen of soil. So they make a lot of stuff to defend themselves from other microbial invaders. So they secrete these compounds with antibiotic and most recently with anti-cancer potential. So a lot of people mine them for their bioactive compounds. So what I do in my standard work, when I look at dynamics across scales and times, I focus on the enzymatic reactions. And I try to print my molecules in 3 dimensions in a flexible fashion where I can show people how this works. And so the enzymes are very flexible in many ways, and they can do these things, and we initiate these reactions by light and see this together. Obviously, I'm here because I want to tell you how light can also manipulate a community of Myxobacteria to form these large multicellular structures, as you see here. And they have homologs in plants. So the same system that I just showed you with the light-regulated enzyme exists in plants, which grow from soil. And so ultimately, this phytochrome regulates also in plants seeding, germination, shade avoidance because it's red and far red light that this phytochrome senses, this photoreceptor. And so in Myxo, people didn't know really why Myxo should have these photoreceptors, and then we showed this link. So our goal is to really study more how light regulates the complexity of myxobacterial development and natural product synthesis. And here, what we stumbled upon as we grow more and more Myxo in my lab, we saw this beautiful patterning. And one of the reasons why I'm here, because I try to model and understand the complexity behavior here. You have to understand that these bacteria are starved, and over course of time, they form these large structures that are highly ordered over course of 14 days. And it's billions of cells coming together in this super ordered fashion in complete darkness. So we leave the plates in the incubator and don't do anything to them, just accept to take pictures. So I made a little GIF with superimposed images over a course of time with these fruiting bodies forming. So we started collaborating with some mathematicians, in particular somebody here at NITMB, to help us understand how bacteria, so many cells under starvation, can do something like this. And what we do know is also if you shine light, you disrupt that behavior. So they will not do this. If you shine light, you can see the circles are not disordered. And we did this in 12-hour light, 12-hour dark cycle. So over 14 days. And on the bottom are just the binary images to help you visualize the contrast between what we see on top. So we can form fruiting bodies on filter paper that we place on top of agar. So we improve the contrast in terms of this. So when we saw this oscillation in the darkness that was so consistent, our immediate question was, could we see some evidence of entrainment, meaning circadian clock in this bacteria? And so I stumbled upon this paper in Genetics Journal on Neurospora crassa, which is a model system for circadian clock. And this is Neurospora crassa under starvation conditions. This is the point of inoculation. And this is the same what we do here with Myxobacteria. We inoculate, and over course of time, when bacteria are starved, they form these beautiful rings that are totally in sync unless you shine light and you disrupt that. So we also wondered whether there is some little bit of nutrients that we see in agar that we dissolve in water that could impact this behavior. So you can buy this ultra pure agar, and if you use the ultra pure agar, or that we call purified agar, we can disrupt also this patterning in the darkness. And so we were able to model, simulate this a little bit with Joe Hibden, who is the director, one of the visiting directors at NITMB. And he was able to visualize this as bacteria move out into the waves to the outer surface and venture across the agar surface. So we have some work that we are trying to model and present. And ultimately, I come here from the point of biochemistry and the work that I've done. So the proteins that sense light, they cannot do this by themselves. They need organic molecules to do this, or chromophores. So this is biliverdin that has to be covalently bound to the protein. And ultimately, over picosecond to second timescales, we were able to look at enzymatic reaction. Then triggers the phenotype of this myxobacteria that we see here that I'm sharing with you. And so the next thing that we are trying to do, because these bacteria are soil bacteria, is really to help and understand their impact on plants and how the plants see the light, right? Because they're the good citizens of soil. How did they go ahead and do this? And so we want to connect our time-resolved work on molecular scale to the microbial communities and ultimately how the plants respond to all of this. And these are my collaborators that I have to thank for all the work. So if you just look up my publications, most of them fall in structural biochemistry field. Mitch Singer is a microbiologist, and Joe Hibden, he couldn't be here today, but he's my mathematician that I go to when I have questions. These are alumni from my lab, and these are my students from a recent meeting. Thank you very much.
So questions. Chinese first.
Hello. Are the observed rings stable or do they move in time?
So they're stable. They appear over course of days. So first we start with one ring, then a second ring, then a third ring, and fourth ring. And moreover, to keep that consistency, to take photos, we have to have multiple plates, because as soon as we take the plate out and image it, we disrupt this phenotype. So then we stop the rings from forming afterwards. So very brief exposure triggers the light-initiated reaction.
Hi, very beautiful talk.
Thank you.
I had a question about the structural part. you're resolving that with cryo-EM?
Yes, and time resolved crystallography. So we have smaller constructs that we visualize. The part that just senses the light, we have crystals of that. And for cryo-EM, we do the full-length protein.
Right. And I was wondering whether do you manage to assemble multiple conformational states for the whole histidine kinase?
Yes, so that's our publication in Science Advances. I couldn't go into detail on it
Okay,
in
sorry.
minutes, but the paper is published. You can see the different distinctions in the light illuminated states of the enzyme. So that's how we know how it moves and the kinase actually rotates.
Okay.
And we just submitted a paper on time resolved cryo-EM. So I'm super excited.
You catch different snapshots as it
Yeah,
how do you
you are limited on millisecond time scale with cryo-EM. So
right.
we just got our first millisecond time point, but we have the dark and light illuminated states of the enzyme. And now we have an intermediate. And yeah.
That's super cool. Thanks a lot.
Thank you. Thank you very much.